Hey Bio Team. So there are two types of cells. There are eukaryotic cells and there are prokaryotic cells. And the difference between them is that eukaryotic cells have inside of them membrane-bound organelles, which is just a fancy way of saying that they have internal compartments that allow specific reactions to occur. So examples of these internal compartments would be things like the nucleus, which contain the molecules needed to build nucleic acids, or the chloroplasts, which allow in molecules needed to do photosynthesis. Whereas prokaryotic cells don't have internal compartmentalization. They don't have smaller membranes inside of them to compartmentalize certain reactions. And so part of the reason that prokaryotic cells have to stay much smaller than eukaryotic cells is so that their biological reactions can still occur. The molecules can still collide into each other. And just a quick note on this prokaryotic diagram. These particular structures inside of the prokaryote might look like membranes, but in a prokaryotic cell, this is just free-floating DNA, one long chromosome looped together, or in this case, it's a very tiny loop of DNA called a plasmid. So how did cells become eukaryotic? Well, biologists attribute eukaryotic cells to a process called endosymbiosis, which occurred when one cell absorbed another cell, and the smaller cell became an organelle. So we'll show an example of that here. We've got this big prokaryotic cell, and we've got this little prokaryotic cell. And the big prokaryotic cell did endocytosis. That is, it absorbed the smaller cell into its cytoplasm, and that smaller cell went on to become an organelle inside of the larger cell. And so now that this larger cell has an internal compartment to it, it has a mini membrane inside of its larger membrane, this whole cell would be considered a eukaryote. As for why a cell would swallow another cell and not completely digest it, not completely break it down, this type of relationship that exists between these two cells now would be mutualism. It would be a symbiotic relationship where each cell is getting something out of the relationship. So this larger cell gets some ATP produced by the smaller cell, and the smaller cell in exchange is protected by the larger cell. So what are the signs that an organelle inside of a eukaryote used to be its own prokaryote that just got swallowed? Well, if we're looking at a organelle inside of a eukaryote, and we see that that organelle has its own DNA and its own ribosomes, that is, it's able to produce its own proteins, that's the sign that it used to be a cell on its own. Likewise, if the organelle inside of the larger cell does its own cell division separate from the rest of the cell, that tells us that it used to be a prokaryote on its own. And last, if the organelle has a double membrane, that is, it has an extra membrane, an extra bilayer, wrapped around its initial bilayer, that tells us that at one point it was potentially absorbed by the larger cell. And so to just show these two membranes here, we've got this one membrane around the organelle that belonged to the original cell, that belonged to the initial prokaryote. But then we've got this second membrane that came from when this organelle was folded into the cell. When endocytosis occurred, an extra membrane got wrapped around the original cell. And that's it. At this point, you guys have some practice problems. We'll see you next class.